So in this video, I would like to present our recent effort in spatial statistics assisted by deep learning and a bit of modeling for spatial temporal mapping of cell organization in tissue. So basically, we developed recently some statistics that are going in two directions, basically level set analysis and clustering. And I would like to explain very briefly how we've used deep learning for that in the context of microglial organization. And this has been a work in collaboration with Theo, Thibault, and David, and recently published in Nature Communication. So, what are the challenges that we've been facing recently? So it has to do with cell classification in the tissue. So suppose you have immunocytochemistry images where you can have access to high resolution images where you see here, for example, glial cells, whatever they are doing. This is not exactly the goal of this presentation. I'm sorry, this is microglia cells. And you can see that they have a variety of shapes from cluster, proliferative, aggregates, amoeboid. So you would like to account for this variety of shapes and be able to recognize as many as possible to see how they are organized in the tissue. So the large variability of shape can be seen here from amoeboid to proliferative. You have here this ramified microglia where you see these protrusions that are very relevant when phagocyting anything in the brain. This is the case of the clustering where they can form really, looks like they are forming aggregates. And again, you continue with this variability of, for example, this migratory where you have these tangential, very non-spherical type shapes. Yeah, and this is another example and so on. So what can we do to be able to account for these cells and in particular to recognize them? So in the context of brain development where you have different post-conceptual weeks here illustrated by these different cartoons, representation of the brain at different stages. So you can have different slices the way that I've just showed to you at those different moments of time. And what are examples of these brain slices, and you would like to not only reconstruct the cells, but also account for how the tissue is going to change across time, and thus this could lead to a change in the tissue itself, and the topology, the organization of the local geometry that has to be accounted for in addition to the clustering of possible clustering of the cells. So we need to identify brain region, and so I'm going to summarize now the approach that we have been developing for the last couple of years, where basically you want to create a pipeline that starts with, as an input, to get these histology images, where you have at refined resolution, you can see very well the shape and the border of the cells. And the first steps, and the first one is actually to create mask for segmentation. That is to go from the cell to create a structure that can remove, for example, elements that are too small to go from. So this is what you can see on the left column is before, and this is after this morphological opening where you remove little small elements. You can also do dilation that will fill the gaps where you see here there was this white little spot that are going to disappear. You can also fill a larger gap, and you can fill filter also by size, so that small size are eliminated, and so you are left with this kind of filter that can be used by a machine learning later on. And this is what you want to do in the next step. So in the next step, you are asking an expert, and that's very relevant in the context of clinical computational biology. So the expert will guarantee that he puts his knowledge in the definition of what is proliferative, amoeboid, ramified, aggregate, phagocytic. So this is an example of 5 different types of cells that you would like to recognize, that is to go from few to thousands, millions or whatever. So you want to upscale, and that's what you want to do with this deep learning approach. That is, you have these cells here, and ultimately the machine learning classification will allow you to get automatically this color identification, where this is given a label to each cell, and thus it's the classification that you want. So in addition to this classification, as I've tried to explain before, you would like also to classify the tissue according to so for example here, the density of nuclei, which allows you to go to start from an image like this and to go to a segmentation. Here we divided the images into 4 regions. I will come back to that, and this is important because you want to do that also across time to see how these regions are actually changing. And finally, this is not enough to have, this is the first step actually, and it's not enough to have done this image classification using deep learning. You would like to extract the statistics and have a full pipeline, so you can go from input to the output. And here we have 2 types of statistics. One is level sets analysis, where you see here that you are interested in generating those lines that gives you what happened in the neighborhood at different distances, and this partition of the space will allow you to look at local correlation between one type of cell with the other type. Another type of statistical analysis is to look at whether cells are clustered. This is represented here in this convex hole, or whether they are inside and forming cluster or isolated. And finally, you would like to understand how this clustering or the statistics evolve over time. So that was the goal of this paper that I'm presenting here today, to have this entire pipeline automatized, and this would help to better classify and clarify the organization, and we applied here on microglia in the human brain. So the first step is brain region segmentation based on nucleus density. So you have your image that you divide into a square, and the teal here will allow you, so we use here the classical cell pulse segmentation algorithm that allows you to count the number of nuclei, and from this you can apply what is called an Otsu thresholding approach, which will, according to this image here, will allow you to give a segmentation based, so you give here a threshold, and you will attribute based on this density of nuclei a label to these different regions that you can see here. So with this, we started with this as an input, and by applying these first parts of the pipeline, so this is at 3 post-conceptual weeks, 17, 19, and 20, what you got in the end is a tissue that was well segmented, that you can see here in 4 regions, where you can recognize the striatum, the neocortex in pink, the cortical boundary in green, and the ganglionic eminence in this orange type color. So once you have this, you can train your algorithm, and for this it's important that you pick up points that are at different locations to guarantee that if there is some kind of heterogeneity, you can have this uniform sampling to get rid of this potential bias. So then you have in this square, you identify your cells, so you apply the mask that I have described before, that will be given to your deep learning algorithm to identify the cells. And so for this, there is a part which has to do with the training of the expert. So we started with the cells, you give the mask, and according to the shape, the expert will give a label. It can be ramified, proliferative, whatever. So he has to click on this button, and when this is done, you have the training. The training is finished, unless you are not satisfied with the mask, and in that case, for a certain finite number of iteration, you can change the mask and start again until the expert agrees that he has a satisfied recognition. Otherwise, you can move on to another cell. So now you can use this training based on a certain number of cells, a few hundreds, with the mask. And of course, there is a certain number of steps. But basically, when you train your algorithm based on 75% of your data sets, you leave 10% for validation and 15% for the test. I'm not going to describe specifically here, but this is a classical unit architecture of your neuronal network. That's what you give as an input, and you have these convolution layers, the max pooling, drop-off, and all of these steps that constitute your deep learning architecture. And then what you get from this cell automated identification, we have these 5 cells. The result of the classification is to give the probability to belong to one of these 5 classes for a given pixel. So if you want to go from one pixel to the entire cells, you have to sum over the pixel, and then you get a probability according to this to belong to one of the class. And so, for example, for this, it's 0.03 to belong, let's say, to the proliferative, 0.47 to belong to amoeboid, and so on. And so finally, you can take the maximum of the probability to assess the final classification. So here we choose this 0.47 and make the classification as amoeboid. And so here I'm just briefly show the classification results. So this is the ground truth from the expert. This is a prediction, and you can see here how the misclassification is represented here for the different class. So we ultimately, when you compute and summarize what you have here, you have a global F1 classification score, which is about 80%, which is quite satisfactory. So you can use also this UMAP representation to show that the 5 classes are well distinguished, showing that the decision to look at this different shape is quite relevant and give different classes, so that we were satisfied. Also, it's true sometimes you see that there are some shapes that could be from one type, one class, and it is found close to another one. But in the majority, this was not the case. So we here ended up with 5 types of microglia. We choose, but it's possible to have others, 3 post-conceptual week, and we had 4 regions. So that's the 4 regions, the 5 microglia state, and one of the first results we obtained was what is the number of microglia of a given type across these different weeks. And you can see here for the striatum, for the ganglion eminence. And what turned out to be a first interesting result is that you can see that, for example, the number is not necessarily increasing. Sometimes it grows, it can also decay, as we're going to see. And you can see that there are sometimes a bifurcation where you have small amount of cells, and suddenly the number of cells is just like multiplied by a significant factor here from 300 to 2000 in just 1 week. So all of these statistics cannot be obtained and can be related to any question of interest. So this is the example of the two. If you look at the neocortex or the cortical boundaries, and the same with the same idea, you can look at how cells evolve across time, and ask, for example, what happened between these 4,000 at post-conceptually 17 to 19, where it's basically diminished by 2. Where are they? Have there been phagocytes? So again, you can start asking all of these questions because you see the result of the statistics. So in summary, we can go from the this immunocytochemistry images to labeling the nucleus that allows to get with this first part of the algorithm and this auto thresholding and some morphological operation on the image itself to obtain 4 regions of interest. And then you can use the machine learning algorithm to segment this microglia into these 4 into this five state, and look at the number of microglia per different regions here that I showed to you above, and again get some very specific statistics. And at the same time, you can look at the microglia class in a given region. So here we choose the ganglion eminence, but in the paper we show also for the other regions, and see what is the total number, which is shown in here, and here we represented in the y-axis the percentage of the cell. So you can see that the amibemoid was predominant, followed by ramified, and then phagocytic. So now we need to go from the general classification to the statistics. And so we started with the cluster statistics. That is, how do you group cells into clusters? Are they really forming clusters? And of course, the notion of cluster depends on the way you are going to look at them, and specifically through an algorithm. So you have to choose something to classify. So here we choose the DBSCAN algorithm, which is an algorithm that will classify according to the number of cells that are around or inside the disk of a given cells. And so if, for example, here you specify that you need 4 points inside a radius epsilon, then all the red cells here, all the red points, are classified as the cluster. And if this one has only one other cells inside, but still at a distance less than epsilon, it is characterized at the border of the cluster. And from this, you can start identifying different clusters. And so you obtain in the end 2 types of cells. Those are isolated and those that are forming clusters. And so this is illustrated here, where we plotted here in the form of a convex hull this cluster. And so the color of the cluster is associated with these 5 different classes. So based on this, you can start looking at the clustering of cells. What is a fraction of microglia that are clustered for the different classes? So you see that, for example, aggregates appears to be very much clustered compared to the other ones, with 70%, followed by phagocytic and ramified, and probably the less clustered is amibemoid. And you can see how this evolved across time. another question of interest is, since you have the indication of the cluster, you can ask what are the amount of cells that are in between clusters. And so this could be important in the possible interactions between the different classes. Here I use the word interaction, although it's not too well defined compared to what is usually understood in physics, but this is a way, again, to look at these specific properties. So you look at the percentage of cluster of type 1 in intersection with cell of type 2. So this is what we did in here. And so you can see, for example, in this graph, what are this fraction of type 1 classified, type 1, type 2, here as illustrated here, how they are, what's the percentage of the cell, and we divided by the total number of cell as shown here in this formula. So again, you can see here, for example, the high number are associated with this proliferative in this phagocytic type. And again, when we looked at these other 4 microglia with respect to ramified, you get this type of evolution with time. So cluster versus isolated, you can look at these for the different region, how the cells are isolated versus clustered in this different region, and how this evolve in time. So again, you can see how isolated can decrease and here, increase, and decrease again for these different regions. So it's not necessarily monotonic. Another statistical approach is given by the level sets based statistics, where you fix a cell of a given type and generate the same way you have this in maps when you look at geographical maps in mountains. So you can see how from a cell like this, the cell is surrounded by microglia of another type. And so this is a very convenient way to look at the local neighbors of a given cells. So here I showed one, but you can see of the other one and see what is the neighbors and how the cells are aggregated around one type of one population. So why is this relevant? Because once you have this level line, and here this is the level sets are labeled from W1 to W9, you can look at how many cells are inside those different regions, and you can compare. Here this is a threshold, which is called the threshold of random threshold. It corresponds to the case of a uniform distribution of cells, and what is below this threshold is actually considered to be randomly distributed. While if it's above, that is, if the number is above this threshold, it means that this is the indication of an accumulation, of a non-random accumulation. And based on this, you can calculate the distance between the one type of cell to the other, and do this over all green cells that are on the tissue. And when you do this and you calculate the distance only on the other type of cells which are above this, you get what is called a coupling distance. And here this was the coupling distance between ramified and aggregate, which was 76 micrometer. So again, what is important is this notion of accumulation score, which gives you the density minus the mean divided by the standard deviation, which has to be larger than this threshold of random distribution. Then you calculate the mean distance according to this pair. And then there is a coupling frequency, which is the percentage of couple of a population. Okay, so this was the mean distance, and so you can fix a population, and look at what is the mean distance with respect to the four others. So this is what we represented here in this form of a tree. And here you have this association frequencies. So more you're close to one, and more this association has been found in this in the statistics of the tissue. And so you can obviously look at how this coupling frequency evolved with time. What is, again, this mean coupling distance also change in time. And this is for this the coupling for the ganglionic eminence that you can see here with the associated coupling distance. So what we found is phagocytic, aggregate, and proliferative are most frequently coupled compared to ramified and amoeboid. So the present approach allows you also to do the statistics over time of the cluster versus isolated. So we look here at the number of cluster cells, the fraction of cluster, and the number of cluster. So we looked also at the fraction of cells in a cluster. So this is represented here. And this colocalization with respect to one type. So here we choose ramified, and you can see how the mean distance in micrometer evolve here with time. So sometimes it can increase, sometimes it is decreasing. We don't have any idea of at the moment of how to interpret all of this data. And this was also you can also do that with respect to the border. So this was with respect to the striatum border. And again, you can look at the different distances of the cell and how they evolve in time. So now I would like to move on to a subject which is not exactly the one I discussed, but I think it's quite relevant because we could use the modeling that we have been developing over the years about trying to understand how the brain is wired for long range, between long range region. And so ultimately you can wire, let's say, from the retina to the lateral geniculus, and from this you can go up to V1. And so the general question that you would like to answer as a computational biologist is how, through the multiple gradients, morphogenetic gradients that you have in the brain, neurons find the target in such a complex environment. And so this is well illustrated here by this case where you have thalamic neurons that have to project into the cortex and needs to go through this corridor. And in the absence of one type of gradient, then they are missing the corridor. And another example is with this thalamo-cortical axon that has to go through this corridor. If this SLIT1 and nitrin are removed instead of being well sorted for these two types of neurons, they are becoming intermingled. So this is to illustrate the complexity of the navigation, and to our knowledge, the combinatorics has never been taken into account to be able to quantify how neurons really find their targets, or at least their places where they have to stop. So for this, we started to account for the fact that the growth cone can be attracted or repelled, and we were also interested in taking into account the fact that you can have multiple gradients that are going to be the relay for moving from one region to another, and ultimately directing the growth cone toward the final location. And so for this, we developed several mathematical models. So this is we illustrated here one where you have a cell which represent the growth cone, that needs to move toward the gradient. So the cells start here, and according to the number of morphogen that hit one of these red windows, the cell can move and ultimately converge towards the gradient. And if you increase, for example, the gradient, you can see how the path is modified. So usually the gradient is generated by diffusion, and the cell motion accounts for the amount of receptors that are being activated in one of these windows, and thus this allows to measure the concentration in one direction, and thus be able to sense whether in one hemisphere of the cell you have more molecule than the other, and in that case you are directed toward this direction. What is interesting is, depending on the flux here, we've shown previously that the cell is able to triangulate where is the position of the source. And for this, we had to introduce a new notion, which is the notion of flux coordinate. That is, the number of particles per unit of time that define the flux is used here to convert the local concentration into a coordinate. And so the position of the source here depends on these fluxes. And so once you have those fluxes, we've shown that with 3 fluxes, you can recover unequivocally the position of the source. And in addition, once you have a cell positioned one place and you have the source, you can use this previous modeling to generate the trajectories. And if you are in the middle, a repellent source, it will be repelled, and you can integrate this into the equation so that the trajectories will avoid this point while converging to this final location. And so this is an example here where if you want to go from one point to the other, and you can do that by 2 means. If at some point you want to avoid to go in the central region, either you add a source which is an attractant, but then if you do this, you are not going to be directed anymore to the second point. So the proper way to find or to give a solution to this is to add here, as you see this purple point, a repellent gradient that will repel the trajectories. So if the trajectory are repelled, it means that they are going to avoid the center, and you can see as the repellent strength increases, you get this arc type behavior for the majority of the trajectories. Okay, and in the end with this, you can generate a path going from where you start to the source. And you can do that by using multiple gradients, and using a memoryless process, the trajectory can be generated from really one point to another. Here this is represented by the different colors of the morphogenes, from one point to another. And so this is interesting in the context of microglia, because microglia can be attracted to the target with respect to sensing the chemoattractant. And so the combination of the two could lead to a new generation of model that will be based on their geometry, because this can be recognized by the machine learning, and see how they evolve in the context of a neuron or glial cells that they have to phagocyte. So in the conclusion, simulation environments are now possible to generate navigation inside a gradient. It's possible to quantify the stopping location viability. So I haven't discussed this, but this will come in a future presentation and study the role of parameters such as the gradient, receptor expression, or brain local organization. The multiple gradient combinatorics remain a challenge, so we are going to provide pretty soon a research work on this, and combined with this artificial intelligence data, it could allow to study the collective migration of one type of cells in the tissue. So the future direction for this deep learning and data modeling is indeed this migration versus proliferation problem. When you have an increase suddenly of microglia in a given region, the question is always, is it due to migration of proliferation? And the statistics here can allow to answer this question by looking at the type of microglia that are actually generated and look at the statistics of the cluster versus non-cluster. There are also interesting extension of this, which is to study this microglia during other type of periods, such as the critical period, to look at how they are involved into shaping the neuronal network. And it's possible also to integrate multi-parameters, such as the calcium dynamics, which give us, for example, if you're looking at a cell, such as the microglia around glial cells, how the local activity can be correlated to the spatial organization. And finally, the classification of pathological tissue could allow us to better understand how these microglia are organized with respect to aggregates. So this is ongoing work that I will present soon. And finally, the simulation and model of navigation. We can now integrate the cell type of shape and to understand how they migrate toward a specific region. On this, I will stop, and I would say that in the next presentation, I will show some of the points that I have presented here. Thanks you for your attention.